Initially, Celera claimed that its material, too, had been derived from 6 anonymous donors, a multicultural group. But in 2002, Craig Venter could not resist letting the world know that the main genome sequenced was actually his own. Today, that sequence is Venter's last remaining connection to the company. Concerned that sequencing genomes, though glamorous and newsworthy, was not proving viable from a business perspective, Celera reinvented itself as a drug company and bid farewell to its founder in 2002. As for Venter, he has established 2 new institutes, one to study the ethical issues raised by modern genetics, and the other to use the genomes of bacteria to find fresh sources of renewable energy. With the whole rough draft in hand, it is now confirmed that there is nothing atypical about the gene density on chromosome 22. If anything, in fact, chromosome 22 with its 545 genes was for its size gene-rich rather than gene-poor. Only 236 genes have been definitively located on chromosome 21, which is about the same size. To date, we have found only about 35,000 genes in total from the entire complement of 24 human chromosomes, 22 plus X plus Y. And while we should stress that the final number can only rise as we make more discoveries, we are all but certain to end well below the 50,000 mark, never mind the 100,000 one. As to how far below, only time will tell. Finding genes is not actually such a straightforward task. Protein coding regions are but strings of A's, T's, G's, and C's, embedded among all the other A's, T's, G's, and C's of the genome. They do not stand out in any obvious way. And remember, only about 2% of the human genome actually codes for proteins. The rest, unflatteringly referred to as junk, is made up of apparently functionless stretches of varying length, many of which occur repeatedly. And junk can even be found strewn within the genes themselves. Studded with non-coding segments, introns, genes can sometimes straddle enormous expanses of DNA. The coding parts, like so many towns, isolated between barren stretches of molecular highway. The longest human gene found so far, dystrophin, in which mutations cause muscular dystrophy, sprawls over some 2.4 million base pairs. Of these, a mere 11,055, 0.5% of the gene, encode the actual protein. The rest consists of the gene's 79 introns. A typical human gene has 8. It is this awkward architecture of the genome that makes gene identification so difficult. But human gene spotting has become less tricky now that the genome of the mouse is better known. The credit goes to evolution. In their functional parts, the human and mouse genomes, like the genomes of all mammals, are remarkably similar, having diverged relatively little over the eons intervening since the 2 species' common ancestor. The junk DNA regions, by contrast, have been evolution's wild frontier. Without natural selection to keep mutation in check, as it does in coding segments, mutations aplenty have accumulated so that there is substantial genetic divergence between the 2 species in these regions. Looking for similarity in sequence between the human and mouse data is therefore an effective way of identifying functional areas, like genes. Identifying human genes has also been facilitated by the completion of a rough draft of the pufferfish genome. Fugu, as it is better known to aficionados of Japanese cuisine, contains a potent neurotoxin. A competent chef removes the poison-containing organs, so dinner should produce only a little numbness in the mouth. But some 80 people die each year from poorly prepared fugu, and the Japanese imperial family is forbidden by law from enjoying this delicacy. More than a decade ago, Sidney Brenner developed a taste for the puffer, at least as an object of genetic inquiry. Its genome, just 1/9 the size of the human one, contains much less junk than ours. Approximately 1/3 of it encodes proteins. Under Brenner's leadership, the Fugu genome rough draft was completed for some $12 million, a genuine bargain by genome sequencing standards. The gene count at present seems to fall between 32,000 and 40,000, in the same ballpark as humans. Interestingly, though, while Fugu genes have roughly the same number of introns as human and mouse genes, the Fugu introns are typically much shorter. Even at 35,000, the present working estimate of the human gene count gives a somewhat exaggerated impression of our essential genetic complexity. Over evolution, certain genes have spun off sets of related ones, resulting in groups of similar genes of like but subtly different function. These so-called gene families originate by accident when, in the course of producing egg or sperm cells, a chunk within a chromosome is inadvertently duplicated, so that there are now 2 copies of a particular gene on that chromosome. As long as one copy continues to function, the other is unchecked by natural selection, free to diverge in whatever direction evolution may choose as mutations accumulate. Occasionally, the mutations will result in the gene acquiring a new function, usually one closely related to that of the original gene. In fact, many of our human genes consist of slight variations upon a relatively few genetic themes. Consider, for example, that 575 of our genes, nearly 2% of our total complement, are responsible for encoding different forms of protein kinase enzymes, chemical messengers that pass signals around the cell. Then there are the 900 human genes underlying your nose's capacity to smell. The proteins encoded are odor receptors, each one recognizing a different smell molecule or class of molecules. Roughly these same 900 genes are present in the mouse as well. But here's the difference. The mouse, having adapted to a mainly nocturnal existence, has greater need of its sense of smell. Natural selection has favored the keener sniffer and kept most of the 900 odor-detecting genes in service. In the human case, however, some 60% of these genes have been allowed to deteriorate over evolution. Presumably, as we became more dependent on sight, we have needed fewer smell receptors, so natural selection did not intervene when mutations caused many of our smell genes to be incapable of producing functioning proteins, making us relatively inept smellers compared with other warm-blooded creatures. How does our gene number compare with that of other organisms? Humans, Homo sapiens, 35,000 genes. Mustard plant, Arabidopsis thaliana, 27,000 genes. Nematode worm, Caenorhabditis elegans, 20,000 genes. Fruit fly, Drosophila melanogaster, 14,000. Baker's yeast, Saccharomyces cerevisiae, 6,000. Gut bacterium, Escherichia coli, 4,000 genes.